Hello, everyone. Thank you very much for attending this Asia Pacific Society of Human Genetics and Hong Kong Genome Institute Distinguished Scholar Series lecture. My name is Brian Chung. I'm the Chief Medical and Scientific Officer of the Hong Kong Genome Institute and also the immediate past president of the Asia Pacific Society of the Human Genetics Society. So today it gives me great pleasure to introduce one of my mentors, Professor Stephen Mann. So Professor Stephen Mann is now visiting Hong Kong, so we have the pleasure of hosting him in the Hong Kong Genome Institute, and he's going to give us a great talk. Let me introduce Professor Stephen Mann. Professor Mann is a professor in the Division of Genetics and Metabolism, Department of Pediatrics, and director of the University of Wisconsin North Center of Excellence for Rare Diseases. Trained in pediatrics and medical genetics, Dr. Mann has held faculty positions at Yale and at U of T, where he co-led the Hospital for Sick Children's Genome Clinic project, which pioneered the use of diagnostic and predictive genomic sequencing in children. Dr. Mann's research interests are broad, from fundamental studies of DNA repair and telomere biology to novel clinical applications of genome sequencing and the ethics of genetic testing. Dr. Mann's current work focuses on discovering new disease genes, developing novel clinical applications for third-generation sequencing, that is long-read sequencing, and advancing the understanding of telomeropathies. He founds the UW, the Undiagnosed Disease Program, and now is developing Badger-Seq, a project to radically improve rapid genome sequencing of newborn infants through the use of AI-driven patient selection and long-read sequencing. So the title of his talk today is Culture Clash, Integrating Precision Genomic Medicine into an American Healthcare System. Dr. Mann, please.
Thank you, Zion, for that very kind introduction. So what I'd like to do today is talk a little bit about bureaucracy, a little bit about medicine, and a bit about technology. And I'm going to tell you a little about my experiences in moving from University of Toronto and the Hospital for Sick Children to a very different environment, a American academic medical center and university, where the balances between science and medicine and clinical care are quite different, and talk about some of our successes and some of our failures. So I want to start by giving you a definition that we have been using for precision medicine. This should be probably pretty familiar to you. The paradigm is that it's what makes a person unique, from their molecular data, their health information, their social circumstances. All of that information and knowledge is integrated into both health research, but also clinical care in order to reduce uncertainty and improve prevention, diagnosis, and treatment. Result being that you improve everyone's health and well-being. The idea is that you can use large data sets. So this is basically big data projects applied to health from multiple sources to gain insights into health and disease so that you can improve the diagnosis and treatment by individualizing your approach to a patient. And it involves researchers and clinicians in a virtual cycle of continual evolution. So if you look at this slide, start with at the very top with patients and families. That's where we start. And you can acquire clinical, environmental, laboratory, and genetic data. Those data then need to be analyzed, and you can generate new scientific knowledge as well as new operational knowledge on the healthcare system side. This leads to proposing new hypotheses and also practice innovations, including implementation. They get piloted. You develop ways to bring that science and that knowledge back to the bedside, and you return that back to families. That's the ideal. You need cooperation from everyone, starting from the bench, moving into the clinic. So when I came to University of Wisconsin, I was asked to start what we call the Center for Precision Medicine. It's a small center at the university. And we were charged with improving the movement of basic scientific knowledge to the clinic. So we really were focused with trying to bring two very large institutions together, the university and the School of Medicine and Public Health. It's a major research university in the United States. It's ranked 8th in overall research funding. And then the university healthcare system is not a single hospital. It's a network of 7 hospitals. It has about 700,000 unique patients and spans a geographic area that is several hundred kilometers in diameter, basically. This graph, basic medical research and then valleys of death, is not original. This has been used a number of times to illustrate the challenges of moving what you see in the basic science area to clinical knowledge and then into clinical practice. And with the difficulties of moving from one peak to another. We were in the middle. We're supposed to catalyze the movement of this knowledge and the application of it, and we're supposed to get the two sides to talk to each other. So that's the challenge. We're in this new building, and we are the 9 faculty. So I came in 2018, and then despite COVID, recruited 8 more faculty. We have people with a variety of different skills. So Irene Aug is a bioinformatics specialist. Mohammed Murtaza is a clinician scientist who focuses on cell-free somatic diagnostics. Irene Kessler is a surgeon who is interested in critical care diagnostics. We have another basic scientist, Galen Hess, whose special interest is developing improved CRISPR targeting. Another clinician scientist, Jeremy Cass, who's an oncologist, who is using organoids to predict responses to cancer. Jessica Lime, another basic scientist, is focusing on epigenomics of cancer. And then Ben Webb, another clinical scientist who is also trained in clinical genetics. And April Hall has a molecular PhD in molecular biology, but also as a genetic counselor. So we have a mix of people. We have programmatic team, biomedical biomarker discoveries. This is really, again, has major translational interest on focusing on functional-based CRISPR screens, epigenomic studies, organoid modeling, multi-omic diagnostics. Dr. Murtaza and Dr. Kisak focusing on cell-free molecular diagnostics, and Irene Hall focusing on using big data to make predictions and diagnoses. And then rare disease diagnoses. So Ben Webb, April Hall, and myself focusing on rare Mendelian disorders. We have a bit of a common technical side. So we have our own long-read sequencing. We have some Illumina sequencing. Then we have educational activities. The usual seminar series, work in progress, an annual symposium. But we also run NORD. This is an American organization, the National Organization for Rare Disease. We run their Center of Excellence. Our undiagnosed disease program is part of the NIH-funded Undiagnosed Disease Network in the United States. We're affiliated with the Undiagnosed Disease Network International. And also there's a regional pediatric genomics consortium that we also belong to. So that's who we are. So how did we do? So I came in 2018. We were tasked with trying to push precision medicine into the healthcare system. UW Health was a healthcare system. So our first push was in oncology. And we chose that in part because there already was a molecular tumor board. And there was a great deal of interest on the part of the institutions, the medical school and the hospital system, in expanding precision oncology. So we run a formal program, genomics-driven precision medicine oncology. We had endorsements from all the leaders. And that was going to involve expanding the existing adult molecular tumor board, which was based at University of Wisconsin. It was already funded by the state of Wisconsin, so we had governmental funding to do that. It was already a successful operation. The next thing was to work on molecular tumor board access for pediatrics or children with cancer. And because of our numbers of how many new pediatric cancer diagnoses we had a year, we felt that it was not efficient to try to stand up our own. So we reached out to sick kids. I had worked on collaborating and setting up the sick kids molecular tumor board and used my contacts there. This was fine with the people in Toronto, my colleagues. And it was also eagerly supported by the oncologists and the geneticists and lab medicine people in the University of Wisconsin. However, we never got it off the ground because we could not identify local funding to pay for it. And so this is it turns out to be a big disappointment. Part of it was who was going to pick up the check, as it were. The university felt very strongly that they did research, not clinical care. And in fact, we as a center were specifically prohibited from using any of our funds to spend on clinical activities. We could spend it on clinical research, but not on clinical activities. The hospital system felt very much that they did clinical care, they didn't do research, and the clinical care had to be paid for. And so it ended up being a standoff, even though it wasn't a lot of money. We were asking to get this off the ground about $80,000 a year for 3 years. And in the end, it fell apart. The other initiative we tried was to stand up a pilot program for using polygenic risk scores to stratify individuals by breast cancer risk. The idea was to develop this with the primary care people so that we would be able to identify those women who would benefit from early intervention and surveillance. Again, lots of support from the clinicians, but it failed because we could not get the hospital to commit resources to protect the time of the primary care clinicians. So again, the problem is that when you're implementing it, even if you have the science of it together, even if you have a good plan and project, you have to have institutions that are willing to commit to providing the resources that are required to get it off the ground. Okay, so that was our initial foray, this precision oncology project. The next one was bigger. It was the University, UW Health's My Precision Health Initiative. So this was meant to be not just oncology, but extending to other cancers of medicine as well. We had an extensive public engagement process. We had multiple town halls. We had committees that worked together to get a consensus from a wide variety of people, including hospital administrators. We engaged the hospital strategic planning people. And despite all of this, we generated some very nice white paper and had a lot of meetings and had endorsement from the dean of the medical school and also from the CEO of the hospital. We then came down to funding. Again, a problem with committing. And after we had endorsement of it, about 6 months later, the message was that the hospital had decided that it really just wanted to focus on precision oncology and specifically on the oncology efforts that were already existed, adult tumor board, and to develop new initiatives as long as they could be relatively self-supportive by clinical revenue. So this comes back to what really drove the hospital system, which was clinical revenue and the prioritization of economic considerations. And so I'll show you one other example. And that was inpatient testing for genetic diseases. So at the University Healthcare System, the UW Health, inpatient genetic testing for rare disease is outsourced. There's not a diagnostic lab in the hospital itself. There is a diagnostic lab at our university, but it is not administratively part of the hospital. The hospital sees that local on-site laboratory as a external laboratory and treats it as such. And one of the problems is that if they are using outside testing laboratories, they have to pay those laboratories for their work. They do not get reimbursed by insurers for genetic tests ordered on inpatients. They get reimbursed if they're outpatients, but not if they are hospitalized. And even though it's not a lot of money per year, it was money that was coming out of the laboratory medicine budget. And this drove, for a period of years, severe restrictions on ordering inpatient genetic tests. They used guidance that had been venerated by Seattle Children's Hospital, and I show you those guidance policy here. Genetic testing for inpatients will only be coordinated, i.e. approved, in exceptional cases. Medically necessary requests would be considered if they could change management of the patient while that patient was still in the hospital. That was key. There was also reasonable expectation the test results would come back prior to discharge. So what it meant, and I'll give you an example, in the nursery, in the intensive care nursery, it meant that we were under ordering genetic tests. This some data here. This nursery admits about 220 infants who are in critical condition a year. We figure that roughly about 25 to 20, almost 30% of those patients will have a rare genetic disorder. You would expect that you would at least order genetic testing on about that many patients a year. In 2020, 19 tests were ordered on 17 patients. Not 60 patients, but 17. 8.5% of the total NICU patients, and just about a third of the patients who we would expect to have a genetic disorder. In 2021, we got a little bit better because we were pressuring, like medicine, almost 12% of the total patients, half of the patients who we've estimated would have had a genetic disorder. After a lot of effort, we managed to get them to lift those restrictions. So in 2024, 56 patients out of 220 had genetic testing. That was about 93% of the people who we were asked to consult on. So the problem with these restrictions, among others, is that misdiagnoses, miss the opportunity to initiate treatment or management decisions while those patients are in the hospital. But it provides, among other things, for a lot of anxiety and moral distress. And I'm just going to mention this is actually a Canadian definition. Moral distress occurs when you feel unable to take what you believe to be an ethically appropriate or right course of action because of institutionalized obstacles. we really felt that that was where situation you were in. Not surprising, it doesn't just apply to healthcare providers; it also applies to parents. Um, when they are stuck in this situation where clinicians are saying this test could help you, but we can't get it. Uh, Postponing testing these children, which is what lab medicine was suggesting, uh, has its own problems, which is if you say, don't worry about it, you can test that child when they come back to you as an outpatient. In the United States, because of the way healthcare is distributed, it's quite uneven. Um, People who are at high risk because of poverty, because of social isolation, don't necessarily come back to the outpatient clinic. Um, and so, as an example, there was a study published a few years ago where almost a quarter of babies who were very low birth weight babies didn't come back to follow-up clinics in the first year of life. Um, Generally, if they were relatively poor, if they were minority groups, if they were far away. Now, in Wisconsin, that's a major problem because it's a very what we call rural state, very low population density. So we have we have 4 sites in the state that have geneticists, the same hospitals where there is a neonatal intensive care unit. That leaves the majority of the state and 14 out of the 19 neonatal intensive care units without geneticists. And in Wisconsin, if you don't have a geneticist on site, the chances of you getting a genetic test in the NICU are extremely low, which means that although we can say, yes, we can order exomes or genomes, they only really get ordered if you are in the centers that have a geneticist. So most of the children who are in a, say, level 3 NICU don't get genetic testing because they don't have easy access to it. And I'll come back to that later. Okay, so just to summarize this part of the talk, what worked and what didn't. So in favor of this succeeding, senior leadership, the dean, the CEO, the senior bureaucracy, all said, yes, we want precision medicine. It's a large academic healthcare system and has a lot of resources. We have a very strong scientific base in the university. However, the institutional culture was not optimal for what we wanted to do. It was not research oriented. And this was a learning experience for me. And one thing that I this quote is actually my own quote. The university's healthcare system, UW Health, their culture was to deliver cutting edge clinical care, but not to invent it. They did not see that as their responsibility. And the university's attitude was, we're here to discover new knowledge, not necessarily to apply it. So we have 2 institutions who are not set up to meet in the middle. And as an example of that, if you look at the healthcare system, less than 1% of the patients were enrolled in any kind of clinical trial compared to other academic healthcare systems that they compare themselves to in the US, where about 4 to 6% of their patients were enrolled in clinical trials. Importantly, they had no dedicated mechanism for funding innovation. This actually was what really sunk us. The hospitals had a single budget system that ran on an annual basis. So if you wanted to do something new, if you wanted to do anything from buy a new centrifuge for the diagnostic lab, or replace the furniture in a nursing station, or stand up a pediatric tumor board, all of those requests went to the same bureaucratic process that happened only once a year. And the rules were that financial return on investment was the primary criteria for deciding whether to approve a project. Uh, that was a major problem for us. Administrative silos were another problem in that every part of the institution, whether it was lab medicine, whether it was primary care, whether it was nursing, has their own budget and they have to be responsible for their own budget losses. So if you ask lab medicine to take on the costs of something like inpatient testing, even though inpatient testing, if you do testing on babies in the NICU, genetic testing is going to save them money, the institution itself, but it's going to cost lab medicine money because it's coming out of their budget. And they are responsible for any deficits. So to them, they have an incentive to say no, even though the institution would benefit. And so we ran into this kind of problem time and time again. So this was a very different experience than when I was in University of Toronto and at Hospital for Sick Children, which itself, the hospital, has a budget of well over $100 million a year to pay for its research activities because it sees as part of its core identity running research. And University of Wisconsin's academic healthcare system did not have that as part of their core identity. And so I think it's part of it is the U.S. system and entirely, and how are things funded. One of the reasons that things work in Toronto is that the primary payer for clinical care, which is the province of Ontario, is willing to spend money on innovation. It's built into their budget. And so they are willing to give money to sick children, to the University of Toronto, to other institutions across the province to pay for and evaluate and implement innovative ideas. That is not the way the U.S. healthcare system works, to nearly the extent it is in parts of Canada. So I'm going to stop talking about bureaucratic frustration. I'm going to talk about I'm done whining. And so let's talk about rare disease genetics. And I'm going to talk about two projects here, our undiagnosed disease programs and a ultra-rapid sequencing program called BadgerSeq. There are a couple of other programs that we also are doing under the rare genetic disease umbrella. Baby Badger, which is an attempt to address what I just mentioned, which is that it's very difficult to get genetic testing in the rural parts of our state, but very uneven access to genetic testing. And also we have this Lord Center of Excellence for Rare Diseases. So let's talk about rare genetic disorders. Now, this should be familiar to you. There are many thousand rare genetic disorders. Some debate as to how many, but we'll say comfortably thousands. They affect somewhere between 6 to 8% of the population, wherever you're looking. And the vast majority of them occur in childhood, maybe 3/4. And many of those are severe enough that about 30% of those patients will die before age 5. Rare disease patients tend to be hospitalized more often than the general population. They tend to stay in the hospital longer because they're sicker. And if you look at specific circumstances like level 4 NICUs, 25-30% of those patients are affected by genetic disorders or birth defects. For pediatrics in the U.S., children with rare genetic disorders account for almost half of inpatient costs. So the medical costs, as a public health burden, rare disease patients costs exceed those for heart disease, cancer, or diabetes in the U.S. So although we think of these as, they're rare diseases, you don't have to pay attention to them from a public policy standpoint. You really do, because in aggregate, they are a significant public health challenge. How do you manage them? Well, I'm going to focus on one aspect, but I think it's a big aspect, which is diagnostics. Diagnosing them is key because if you have a diagnosis, you can understand the prognosis, you can optimize treatment, you can estimate recurrence risks to families. And many patients who have a rare genetic disorder in the United States are never evaluated for genetic etiology. And when they are evaluated, these diagnostic odysseys can take years. So you can have a child who is noted to have some problem when they're at birth or in infancy. It can take 5 or 6 years, very key 5 or 6 years in early childhood to sort it out. And that evaluation can be quite invasive uh, and costly. And most of the patients who end up having a rare genetic disorder are initially given the wrong diagnosis. And speaking of diagnoses, undiagnosed genetic disorders are the most common ones. And less than 50% of our patients will receive a definitive diagnosis. And the people who are in that undiagnosed group, we recognize they have some rare disorder, but we don't know which one. Because we can't give them a label, they have a difficulty getting access to healthcare and to social services. There's additional anxiety and stress because they have a black box instead of a diagnosis. They don't know what to do. And there's a lack of community support groups. If you have a diagnosis, there may be a family support group focusing on your particular diagnosis, but not if you don't have a diagnosis yet. Now, it's not that we haven't tried to improve diagnosis. We have. And if you think about historically, when you started out with using genome-wide methods, first genome-wide methods for diagnosis were karyotypes. We were doing that starting in the 1960s. However, the diagnostic rates were lousy, 3 to 5%. Microarrays were much better, 10 to 13%. So, going up from 3 to 5 to 10 to 13 was a big jump. We were all happy about that, but it still was a very small minority. Exome sequencing and then later genome sequencing have gotten our diagnostic rates up to past mid-30s, maybe 40% or so for trio short-read genomes. And so we're quite happy, those of us who have been in the field a long time, because this is much better than it used to be. But we're still less than 50% for genome sequencing. So why is that? Well, there are a number of issues. First of all, short-read genomes only cover about 93% of the genome because there are regions that are just not easily analyzable because of pseudogenes and repetitive sequences. There are also certain kinds of variants that are not easily assessed, structural variants, repeat expansions, as an example. And then when you think about the reference sequence that we most often use for diagnostics, which used to be 37, but now is 38, is incomplete. It's been estimated to miss about 15% of known variants and about 8% of the genome. We also know that the 38 reference genome and NOMAD, as it's currently constructed, are not truly representative of genetic variation in humans. And I didn't put a slide that illustrates the NOMAD situation that I didn't put in this talk because I'm trying to keep it short. But it's remarkable how uneven the coverage is in terms of ancestral background in NOMAD. And in particular, both East Asian and South Asian populations are way underrepresented. It's gotten better, but it's still a major issue, and that it's not representative. And another challenge is that it's short read data. So if you are doing long read sequencing, you don't have a good reference for making sense of the structural variants that you found in long read data. So there are a number of weaknesses that we currently are dealing with. And for a variety of reasons, we can't classify most genomic variants in terms of their pathogenicity. And attempts to improve that by, say, doing functional analyses have been made very slow progress because they're very low throughput traditionally. And the ability to do massive parallel assessment of variants, although attractive in theory, has limitations in terms of what genes you can assess with massive parallel approaches. There have been some very good examples, like BRCA1 and 2, for example. But if you don't have a phenotype that you can assess on a single cell level, it's going to be very difficult to push that kind of functional assessment forward. Um, and there are lots of genes that we still think we haven't found yet. Um, okay, so what have we been doing? When I arrived, I started to set up an undiagnosed disease program. We got sidetracked by COVID. That slowed us down a little bit, but we did get it up and running. And the goal was to focus on trying to provide answers for patients who had been through standard clinical evaluations, improve our knowledge of the pathobiology, and discover some new disease genes. We have a very, the workflow should look fairly familiar in terms of undiagnosed disease program. We take in referrals. We do some publicity, but we look at the cases that are referred. We have a case review committee, and we either decide to accept. If we don't accept, it may be that we have recommendations in terms of what the referring clinicians could do instead of seeing the patients. If we give them the green light and we see them, we review available data, including sequencing data. That may result in reanalyzing the sequence data, typically exome data. If not, then we will go on to do genome sequencing. We've been doing a mix of either short read or long read. We do this in our research laboratories setting. If we find variants that we think are clinically relevant, we then validate them in a CLIA laboratory, which generates a clinical report, which we can then return to the participants. If we do genome sequencing and we don't find an answer, we have some other tools we can move on to, including RNA-seq and biochemical analysis and sometimes functional analysis. This is some older data, but in the first, when we first started up, as of the end of December 2024, we had 258 referrals. Majority were coming from genetics. We were hoping to get more referrals for non-genetic specialists, but mostly from genetics. A mix of adults and children, about 2/3 children. And we ended up taking about half of our referrals. And the vast majority had prior exome sequencing. And in the last year, it's been in the mid-90s% of people who already had prior exomes. We classify our referrals by our level of interest. We can't tip on everyone who is referred to us. So we prioritize situations where we think we have a high probability of finding an answer. So if there are multiple affected family members with an interesting phenotype, they're our first priority. If it's a complex or rare phenotype, they're second. If it's a common phenotype of interest, but we think that it's solvable, that's next. If they don't meet our eligibility criteria, we say no. And sometimes they're enrolled in another study already, so we also say no. Most common phenotype is someone who has neurologic abnormalities, which should be no surprise, and developmental delay in behavior is the next most common. So between the two of them, the majority of our patients have some, one or the other or both. We have also seen patients who have primarily cardiac problems or GI or immune, some are ophthalmologic. Okay. I mentioned a little bit about what's in our toolbox. So reanalyzing existing data, short read genomes, long read genomes, RNA-seq. We've done some optical mapping. We've done some functional analysis. And The right-hand side of the slide is just a little bit of an advertisement for long-read sequencing. For practical reasons, we've settled on nanopore sequencing rather than PacBio. And the reason why is one of scale. Um, to invest in PacBio, you have to buy a very expensive machine, and you have to keep it running constantly in order to reap the benefits. And we were not in that position. We didn't have enough work, and we didn't have enough budget. And so the attraction of nanopore sequencing was that you could, for substantially less money, acquire a machine and run productively run intermittent sequencing samples. So, if we're running 2 or 3 genomes a week, that's fine. It wouldn't make sense from a financial standpoint to buy a PacBio sequel to do that. Um, I'm not sure what the level of expertise is about nanopore sequencing in the room. I'll assume that you know a fair amount about it. So when we first started this, the idea of getting a nanopore sequencer, we had a lot of opposition of, oh, it's too error prone. Performance isn't good enough. That is in 2024 with the new flow cells, the 10.4 flow cells and improvements in base calling, that's pretty much gone away. And so we're no longer getting people complaining about the error rate in our nanopore data. But we're actually quite happy about that. I'd be happy to talk more if somebody wanted to talk about the nitty-gritty of running the ONT platform. At one point, we were planning on switching to nanopore as our primary tool, but it's not really cost-effective at this point. If someone gifted us a lot of money, we would switch to Lottery only. But we're not in that position, so we're still using it selectively. So in the first 44 cases that we analyzed, we saw 15 of them. And I'm not going to dwell too much on this slide other than to point out that, not surprisingly, undiagnosed disease programs, if you take on all your referrals, you're going to get a very wide mix of results when you do make a diagnosis, when you do solve a case. So that includes well-known disorders. It also includes newly described disorders. It's not on this list, but in our first 50, 60 patients, we found 2 RNU4-2 patients, which when we first found it, it was like a month after the paper had been published. So I was like, oh, this is cool. But then we found another one shortly thereafter. So now we realize this is just going to be super common. So I'm not going to talk about that one, but I will show you just a few examples. So this is a case where long-read sequencing helped us. So this was a pair of siblings, a 4-year-old girl and a 3-year-old boy. They both had moderate global developmental delays. They were hypotonics. They had some dysmorphology, large heads, frontal bossing, mild synophorus. The girl had a congenital eye movement disorder, and the boy had a occult submucous cleft height. They had a lot of genetic analyses. This 4-year-old girl had some panels, a spinal muscular atrophy panel, which was normal. Microarray was normal. The cerebral palsy panel, which found a lot of VUSs. Neuromuscular disorders panel found some more VUSs. And finally, they did a quad exome, which fortunately found all the previously identified VUSs. So that was good. Quality control there. And also found a parental variant, a frameshift variant in the KIA-586 gene, which you might know is Joubert syndrome. The 3-year-old brother also was part of the quad exome, and they found the same paternal KIAA variant. So what about the previous testing? There was a maternal pathogenic COQ8A variant It's associated with ataxia, seizures, and developmental delay, and both of them had it. But it was recessive, so one allele, not the other. There is a paternal AGR in the US. Again, autosomal recessive, and only the sister had it. It was for a neuromuscular issues. There was a maternal AMPD1 variant. Again, autosomal recessive. It was only a myopathy, not developmental issues. There was a paternal MBAS variant, autosomal recessive again, with short stature and optic nerve atrophy. Again, not a good fit. And then a maternal PCCA VUS, propionic acidemia, only affecting the sister. So those didn't, for various reasons, make a good story. The last one was this paternal KIA-0586 variant. This for autosomal recessive Joubert syndrome. Both of them had it. So which one would we pursue first? Well, in thinking about them, we were attracted to looking at the Joubert syndrome variant, in part because they both had it. It wasn't a VUS, it was a frame shift. And there was a way we could do some reverse phenotyping because Joubert has some very distinctive features, including MRI finding. So classically, people with Joubert syndrome have an MRI finding called a molar tooth sign. And so we got an MRI on first the girl, and sure enough, you can see in the middle of that MRI image something that looks like a tooth. So molar tooth sign, positive. We also incidentally found out why she had this problem with her left eye. She had an absent left sixth cranial nerve, so we explained that one as long as we were at it. But then we also imaged the boy, and he had a molar tooth sign too. So together, they met the clinical criteria for Joubert syndrome. So now we had a clinical diagnosis, but we were still missing that variant. And KIAA is not the only genetic cause for Joubert. So was this actually causal or not, or are they just carriers of it? So we did long read sequencing, and we found that there was a maternal 8.2 kb deletion that spanned exons 8 to 10. You can see that this is the quad. You can see the sister, you brought in, and the mother all have this deletion. There were reads that spanned the deletion, all three of them, and dad does not have it. So we now had our diagnosis. The clinically met the criteria for Joubert, and we had a situation where both affected individuals, both kids, had with compound heterozygotes or pathogenic variants in the Joubert gene. And we went back and looked, they had an exon, right? And this deletion flipped off exons 8 to 10. So could you see it in the exome data? Well, here it is. So this is the data from the testing lab, GDX. And you can see here, this is exon 8, 9, and 10. Here is dad, here is mom. Mom is supposed to have the deletion, as are the two siblings. So it would be very difficult, I think, to call by read depth. But these are hemizygous, and that's homozygous. And so I could see why they didn't call it. But it was there. Okay, so I'll give you another example. This is a 18-year-old girl that was referred to us because she had a complex history that included neurodevelopmental problems, global delays, severe intellectual disability, autism, hypotonia. In face of the normal MRI, she had immunodeficiency as well, low B cell numbers, hypogammaglobulinemia. She also had some GI problems, feeding intolerance, dysphagia. She had some other medical issues, scoliosis, neurogenic bladder, hydronephrosis, and she was dysmorphic. Sparse hair, alopecia, prominent eyes, a small nose, odd mouth, multiple pupil fissures. Only one in her family. So the initial testing, which was way back in 2006, she had a karyotype when she was a baby. It was normal. Subsequently, 4 years later, she had a microarray, and that found a 214 kb deletion on chromosome 16p11. Now, 16p11.2 is a hotspot for deletions and duplications. Most commonly, they involve breakpoints called breakpoint cluster 4 and 5 or 2 and 3. This turned out to be a 2 and 3 deletion. And that clips off a number of genes. It's associated with a mild to moderate phenotypes of autism, macrocephaly, obesity, seizures, hypertonia, but not dysmorphic. Remember, it's just dysmorphic. So here we have a genetic diagnosis. Well, it turns out that the geneticist who was taking care of this child wasn't satisfied with that diagnosis, didn't think it explained enough. So when exome became available, they arranged for an exome, which was negative. In 2021, they arranged for a reanalysis. He was not willing to give up. And this time there were 4 VUSs, including a VUS in JAK1. When we got the patient, we reanalyzed the data and we decided that the JAK1 variant could be reclassified as pathogenic because, among other things, earlier that year, there was a paper published that showed that that particular variant had been found in families that had a dysfunctional immune system in 6 affected individuals in 3 families, and that variant was functionally abnormal in vitro. So now we had some reasonable data that that variant was not benign. So we could say we now have both this deletion on 16p11, but we also have a JAK1 pathogenic variant, which could explain the immune dysfunction that she had. So now we have another piece of the puzzle. We also did short-read genome sequencing, and we found a de novo SMARCA2 deletion, which you can see on the left-hand side, just from the read dots. You can see it's there. It's 26 kb de novo deletion, not present in either parent. And again, looking through the literature, we found a prior case of a patient with a 32 kb deletion, which was affecting the same exons of the gene, 20 to 26. Now, SMARCA2 encodes for DRM, which is part of the SMI-SNF complex of primitive remodelers. And it's associated with 2 different related syndromes: Nicolaitis-Bärder syndrome and blepharophimosis-impaired intellectual disability syndrome. And it turns out that the string of variants that give rise to one versus the other are somewhat dissimilar in terms of where in the gene you find these. And recently, there was a paper that documented that there are distinct DNA methylation signatures. Remember, this is a primitive remodeling. So it's not unreasonable to think that it's going to affect methylation and epigenetic marks. And so we now have a way to answer one of the other questions we had, which was, we have a deletion in SMARCA2. That could explain the dysmorphology and some of the rest of the phenotype, but is it Nicolaitis-Bärder or is it blepharophimosis syndrome? And we can use methylation analysis to tell the difference. So together, the 16p deletion, the JAK1 variant, and the SMARCA2 deletion explain this child's complex phenotype. It also adds evidence that JAK1 gain-of-function variants, which is what that was, can cause clinical immune disruption. And that we could find structural variants that were missed by microarray and exome sequencing. I do have a comment here. We use something called Occam's razor all the time in medicine. The idea being that as a general rule, you should look for the simplest single explanation for the problem at hand. That's very useful in diagnosis. You don't want to postulate too many causes, but sometimes you have to postulate more than one. This is one of those cases. And the other thing is that getting a diagnosis in this patient was not just a matter of our doing our work, it was a matter of the clinician not giving up, not stopping at that first diagnosis. That was just, I think, an important take-home lesson for clinicians. If your test results, if your exome, if your microarray, whatever, if it doesn't fully explain what the patient, then you should think further. Because if you think about it, what are the chances of a person having not just one rare disease, but two rare diseases? If you think that a rare genetic disease is, say, 6% of the time, then if you've got one rare genetic disorder, chances are 6% that that person will have another one. So it's not that uncommon if you're looking at if your job is to look at patients with rare diseases, you're going to come across patients who have more than one, and you shouldn't be surprised. Okay, here's one other example. This is an 8-year-old boy, moderate developmental delays, delayed motor function, hypotonia, obesity, dysmorphic, um, had a microarray, found a 2.7 megabase deletion on chromosome 2. Exome sequencing found the same thing. That didn't seem like a good explanation. So they did a karyotype, old-fashioned genomic analysis, and they found 4 abnormal derivative chromosomes involving 2, 3, 8, and 21. The problem was the karyotype didn't really tell us very much in terms of what these were. And so what we ended up doing was to do long-read sequencing and optical mapping. I'm not going to go into a lot of the details, but to show you that one of the things we could do with the long-read sequencing was to map the breakpoints and to sort out what was happening. So on each of these slides I'm going to show you the blow-up of the karyotype. This is chromosome 3 and the derivative 3. And this derivative 3 has a part of chromosome 2 on it. And with the long-read sequencing, we can map exactly where those breakpoints are down to the nickel. And we did the same for the other chromosome 3 breakpoints. We then so this was the chromosome 2 breakpoint. This is the chromosome 3q 11.2 breakpoint. This is a 3q25 breakpoint. All in all, we found 10 breakpoints, and when you look, most of them had very small nucleotide gains or losses, suggesting that what we were looking at was some homologous end joining events. And what we were hoping for in this complex series of rearrangements was that we would have interrupted or deleted genes. But the answer is we didn't. This is where we're stuck. We're so proud of ourselves. We did genome sequencing, we did long read, we did optical mapping, we sorted out all the breakpoints. They were far more complex than the karyotype suggested. We had we had inversion, we had these 4 chromosome translocations, but the genes that were disrupted were not genes known to be associated with a phenotype. We even looked at genes that were near the breakpoint because we thought, well, if you're interrupting a local CAD or you might be interrupting the connection between a long-range enhancer element and the coding sequence, we haven't been able to establish that yet. So we're still looking. Okay, so end of the UDP story. We're solving about a third of our cases. And since these are almost all exome negative cases, we're pretty happy about that. That was higher rate than we thought of because we're only dealing with cases that have already been through a clinical exome. So why are we doing better than the exome? Some of it is time. Some of the exomes were done several years ago and new genes have been discovered. So that was sort of easy to do, like the RNU4-2, not on the exome. We found them on genomes. Sometimes it's just that there were variants that couldn't be picked up on exome. Sometimes we were just lucky. But we are able to push that so that cumulatively between a clinical exome and our reassessing the patient, we're able to push the cumulative molecular diagnostic rate past 50%, which is good. Future directions, we're trying to expand. We're trying to take advantage of nano-sequencing. And in doing so, we're looking towards taking some of our unsolved cases, doing de novo assembly of the long read data and alignment to T to T. And we recently joined the NIH UDN, so we're trying to take advantage of that and collaboration with other UDN members. And finally, sustainable funding models. That's critical for all undiagnosed disease programs. We're in this typical boat of we launched it, we have been living on our initial funding, but we have to find sustainable funding. So I'm going to finish by talking about the NICU project. It's very briefly. Again, probably know some of this. Babies in level 4 NICUs have a high chance of having a rare genetic disorder, and that chance is about 25% or higher. We know that rapid or ultra-rapid sequencing can be very important for these children. You know that the faster you make that diagnosis, the greater the impact on care and costs. You don't have a lot of time because NICU stays averages about 2 to 3 weeks. And we wanted to address 3 problems. One of which is that not everybody gets it. I already alluded to in Wisconsin, the majority of patients who are in NICUs don't have a geneticist and they don't get exome to genomes. The current technology misses important variants, like you don't get oscillation analysis, so you don't miss things like greater load in the newborn period. And speed, it takes too long. I'll just give you an example. This is our own data from our own hospital. Currently, it takes almost more than 2 weeks, almost 3 weeks, from the time of admission to the NICU to the time of the diagnosis, molecular diagnosis coming back. So why is it that long? Part of it is it takes so long to get the test run that you send it out, it has to go over to the testing lab by FedEx, that takes a day, and then they give you a 7 to 10 day turnaround time. But the other part of it is it takes a long time to decide to sequence that baby. So from the time of admission to the time of consult can take more than a week. So we want to shrink the total time. So we want to work on both problems. And we want to get that down to 3 to 5 days from its current 2.5 weeks. So the idea is to address both issues. The time to identify who to sequence, we want to address by turning it around. Rather than having a genetic consult first before you decide to sequence, decide who to sequence first and get the consult later. And so the idea there is to analyze data that are in the electronic health record and rank patients based on the data in the record. Take those patients who are above a threshold and initiate both the genetic consult, but also initiate sequencing. So you start the sequencing before you have the formal genetic consult. And we think we can do that within about the first 24 to 48 hours after admission. At the same time, don't send our sequencing out. Do it locally, and do it locally on our nanopore sequencer and get that done in 48 hours from the time of the blood sample to the time of the return of the initial analysis by sequencing using 2 flow cells per snout. So the actual time on the sequencer is 36 hours. I'm going to tell you a little bit about our experience with the AI-assisted collection. The idea is you're going to text mine the electronic medical record of all the NICU patients every night for human phenotype ontology terms. And the text mining is using a program that was developed by the same people who are maintaining the HKGI database. And I'm just giving you an example at the bottom of the slide. This is a paragraph describing a patient who actually has Fanconi and it's a standard sort of clinical summary. You could use that software, FastHBOCR, to extract HBO terms, shown on the right, from this clinical record. Those HBO terms are computer friendly and can be used to characterize the patient. We feed those terms into another piece of software called the Mendelian Phenotype Search Engine, developed by Mark Yandell at University of Utah. And what it does is it was trained to mimic the decisions that clinical geneticists make when they decide which patients in NICU to sequence. So that's all it's doing. It's just imitating the geneticist decisions. And it calculates a log likelihood ratio. You can see a plot of those ratios. This is their published data. These are the kids who didn't get sequenced and their scores. This is the distribution of scores of those patients who were sequenced by the geneticist, who's in pink. And you can see that there's some overlap, but there's a clear tail of patients who have OPSC scores that are high enough as they would have been picked by geneticists for sequencing. But that's what they were able to do in Utah and in California. We needed to bring that in-house and to see how well it would work on patients. So we did a retrospective study where we took 356 babies who were admitted to our NICU, and we set up a system where we took their data and split it into a test set and a training and validation set, and ran this software, optimized it, knowing that of those 356 patients, most of them didn't get a genetics referral. 66 had a genetics referral placed for the first time after admission. And those are the patients who we're most interested in, because those are the patients for which our genetics team had to make the decisions. So we were interested in training using our data. And what we found was it worked pretty well. So this is the performance. This is the operator curve, and we can get AUCs that are in the mid 0.8s, which is pretty good given the prior probability is about 25%. This is a distribution of our scores of patients who weren't sequenced and patients who were, which were pretty satisfying. What's in practice? It means that software was good at imitating the decisions, but not only is half of what we needed the software to work faster than clinicians. Let's show on this one. So if you looked at the patients in that group who were seen by geneticists for the first time in the NICU and had underwent exome or genome sequencing, there were 43 of them. If we look at what the model predicted using only the day of admission notes, it predicted 37 of the 43. And at day one, only 9 of those patients had been chosen by sequencing by the clinicians. By day 2, it had gone up to 17. The software had picked another 2. By day 5, software is sort of maxed out at 40 out of 43. They were still at 23. And then by a week later, the clinicians still hadn't had a lot of patients they hadn't picked yet. But the software had plateaued out at 40. So the software is working much faster. 86% of the patients who eventually underwent exome or genome sequencing were picked by the software on day 1, versus only 20% by the clinicians. And even by 5 days, 93% were picked by the software and only 55% by the clinicians. So that was a key thing we needed to be able to show, was that the software worked faster, significantly faster. That's the software side. Happy to talk more about that later. And then the sequencing side, just very much just tell you about the actual workflow. We allow ourselves 36 hours to do this actual sequencing. We do it on a P24 median. We use 2 flow cells per genome so we can harvest the data at 36 hours and still get more than 30x read depth. That means you're using 6 flow cells per trio. They're using the super high accuracy base caller with alignment on the fly. And then we use a fairly standard epitome workflow for human genomics. We're saying AI-assisted tertiary variant analysis. Now, we initially used Fabric software, commercial software, but we are now test driving software from other companies. So we may actually change that. But all in all, we're giving ourselves 48 hours from blood to the final assessment. I know I'm running late, so I'm just going to tell you, we did our first trial using that workflow, using trios from our undiagnosed disease program, 3 positive controls and 6 undiagnosed. And we averaged 3 to 4 hours for the library prep time, an average of 112 gigabytes of data in 36 hours, 7.3 KB N50s in terms of read length. We were able to do our assembly and variant calling of about 4 hours of time on the compute, and then uploading the data to Fabric with another hour and a half, and they took about 2 hours. So we were able to make our time. I'm just going to summarize there so we have some time to chat. So we're able to do our text mining of the electronic medical records, our HBO analyses. I didn't tell you, but we do this in a HIPAA, privacy protected computing space. The large majority of patients for sequencing we could identify on the day 1 of admission notes. And our goal of sequencing from DNA to variant analysis in 48 hours is realistic. ONT platform is well suited for what we needed. We needed intermittent, moderate volume sequencing. You couldn't spend a flow cell, we couldn't spend a Illumina flow cell on what would amount to 3 genomes. But we could spend 6 flow cells on the Prometheans. What we're doing now, we're trying to improve the MPSC model performance, among other things, by giving it more data to train on. We're fully automating the text mining and the MPSC analysis using log data. We're trialing other tertiary analysis software, Imaging, Fabric, Lucid, Sequon, and GeneX. And for the long term, we want to do better than these just using HBO films. We think that we're leaving information on the table if you don't also include laboratory data and imaging data to see if that could allow us better performance. So I'm going to stop there and acknowledge the people who are involved in the undiagnosed disease program, the Badger Seek project. I love our patients and families. Consider it nice and their funding.
Thanks a lot, Dr. Mai. Wonderful lecture. And just like all great movies, it's always at the most interesting part. Then, okay, we will wait for a sequel of the movie. This is a very exciting work. Dr. Mai, I like the talk very much, especially because you're so generous to share also on the difficulties and the problems to do to implement genomics medicine in a real life situation. You have so many other barriers like the system, like the policies, like the finance. This is really helpful. And I think Dr. G and I echoed a lot on also happened in this part of the world. I think it is really a big pity, especially on the pediatric molecular tumor board, because if it is allowed to run a longer period of time, you will be seeing a very different pattern and what would be the area that is most useful in pediatrics. I guess I have heard talks that said sometimes the genomics diagnosis is more accurate than what was provided by capabilities. Do you see that too?
No, it is interesting. My experience really has been with the project in Toronto on the KICS. It's called KICS, KICS. Um, and most of that work was done by Adam Schleim. And Adam has been using RNA-seq and expression analysis to profile tumors. And he's found that they have, he's not the only one, but it turns out that you can characterize the tumors by their transcriptional signature. Now, you think about a generation ago or 2 generations ago, people were, it was the era of histopathology and immunohistology, and you could characterize tumors by their appearance under the microscope. And this revolutionized tumor classification. But now we're able to use molecular analyses, and by doing molecular analysis, you can confirm the histologic diagnosis in the majority of cases, but not always. There are cases where it's clear that you can make a diagnosis by molecular signature, but you couldn't make by histopathology. So you can clarify tumors that are uncategorized. And it also points out something that is useful from a therapeutic standpoint and from a basic biological standpoint in finding similarities in the transcriptional profile between tumors of very different origin. If they have similar transcriptional profiles, that suggests that they have some of the same commonalities in terms of what their pathology is, and therefore what might be therapeutic approaches to treating those tumors. So it's been very useful.
I think that I think we have a lot of to learn from you regarding the long way rapid sequencing you just talked about. It's very amazing. So I think maybe I would just ask one question to as a warm up. We are very impressed by the AI assisted screening of patients who are eligible for the those whole genome sequencing. But because we also did a lot of work here in HKGI, what we face is that we cannot quite recap the performance that the paper is suggesting, the AUC of 0.6. We are guessing maybe that's due to the different in format of the electronic health record. So I'm not quite sure. Maybe in the state are the electronic health record unified so that you won't have much problem to to bring the tooth develop in California to Wisconsin. Or you also have a very different style of electronic health record that you optimize actually to make it fit into your system.
So most electronic health records in the United States and in Canada are basically run by software sold by 2 companies, either Epic or Cerner, with Epic having the more market share. And so the systems at Utah and at San Diego Rady Children's, which is where MPSC was first trained, are Epic sites, and we're an Epic site as well. In theory, because these are clinical text notes, they're not, they're free-form data that the clinicians entered. I don't think it should make that much difference whether it's Epic or Cerner. On the other hand, you could say that these are mainly neonatologists' notes. The neonatologists in North America seem to have been trained in a very similar way, so they generate very similar notes. That I'm speculating as to why you might see a difference in performance. But we were worried about whether a model built on one set of clinicians would translate to another set of clinicians. And in our case, it seems to have done reasonably well. I still think that I'll make two comments. I'll repeat the comment I made, which was, I think we are leaving information on the table by only using FlinPubs and only extracting HBO terms in FlinPubs. There's more data there. And so you ought to, in theory, improve the models. Now, the other fault with this model is that our goal through this, the MPSC, is to be as good as a clinical geneticist. I'm a clinical geneticist. I know I am not perfect. We cannot, for certain, predict who in the nursery is going to have a genetic disease. So can we do better than that? One way would be to train the model on a set of data where you've had genomic sequence on every baby in the NICU. So you didn't have to guess. And so you wouldn't train on the decisions the geneticists made, you would train on who had a diagnosable by genome sequencing disease. Now, we haven't been able to do that yet. There are barriers both in terms of it would be more costly to sequence everybody. It would be a major leap to get an institutional review board to approve a protocol like that, where you're basically saying, I want to sequence patients who I'm pretty certain will not have a genetic disorder. The majority of them wouldn't if we sequenced everyone. And the last thing is try to convince parents to agree to it. That's also a problem. So we're addressing that by working on a proposal where we take retrospective data. So a retrospective cohort of babies in the NICU, all babies for a certain period of time. We sequence residual lab samples, and we have what's called an honest broker. This is a position that already exists at our hospital, where they take the clinical data and they anonymize it before they let us see it. So we would get the anonymized clinical data. We have the sequence for everybody. We have all the patient's data. We can now do that study retrospectively in terms of who would have had a genetic disorder and who didn't. And we would have the clinic notes in the clinical record to train the model. So that's our way, our attempt to get around it. Because we know that if we ask for consenting, about a third of the, even on the best of days, about a third of the parents would say no. We don't want to confound our analysis by missing a third of the cohort.
Any other questions?
Wait, do you have extra questions?
Sure. Sure. I have a privilege to ask another question. So, in our hand, actually, I saw that you use what we call the super accuracy scaling model. On our hand, we actually evaluate that a lot. And what we see is that yes, with the super accuracy calling model, we improve a lot in many regions in the genome, but we still see quite a lot of false positives or even false negatives around the homopolymers. So we struggle a lot to address those because we also offer rapid genome sequencing to the MSU patients. So basically, it's very often we encounter those where we cannot decide whether it's a true positive or we need further follow up. So may I ask, in your hands, how do you handle those?
We do not have I don't have a magic workaround for that. That is still the major weakness. I think Nanopore themselves is trying to spin that problem away by saying, oh, there are not that many homopolymers in medically important genes. And all these and that, that's their response, I think. But it's not been solved. It is better, but it's clearly full.
Okay, if we don't have further questions, may I ask you to give a round of applause we talk about for the excellent job. Very
Hopefully we'll discuss again tomorrow for closely discussion on interesting projects we shared common interests. All right. Thank you very much.